After looking at an overview of cellular respiration in the previous lessons, now we're going to take a closer look, and we're going to start here with glycolysis. Here are the objectives. Now, glycolysis is an evolutionarily old process that occurs in the cytoplasm. So it doesn't occur in the mitochondrion. To give you an overview over here, I've got the stages of cellular respiration. Again, we looked at this as glycolysis, this as the Krebs cycle, and this down here as the electron transport chain. And so we're focusing on this step, and I said that it occurs in the cytoplasm. So we're not actually in the mitochondrion yet, we're out here in the cytoplasm. It's evolutionarily old, and it takes place in both eukaryotes and prokaryotes, or both eukaryotes and prokaryotes carry out glycolysis. The name glycolysis, if we divide it up as glycolysis, tells us what it means. Lysis means splits. Glyco is for glucose. So this is a process of splitting glucose. Glycolysis splits glucose, and as we'll see, it takes the 6-carbon glucose. Here's a hexagon to represent my 6-carbon glucose, and it's going to split that into 2 3-carbon molecules, each of which is called pyruvate. So pyruvate is a 3-carbon molecule. So glycolysis is the process of splitting the 6-carbon glucose into 2 3-carbon pyruvate molecules. And although we're going into more detail here than in previous lessons, I'm not going to show all of the many reactions that are happening here. I'm just symbolizing them with these overview arrows. But keep in mind that they are enzyme catalyzed reactions, and that's important because we can turn up and turn down the activity of enzymes, for example, by feedback inhibition, and that allows fine control over the activity or the speed of glycolysis. If you need more energy released, say, from stored glucose, then you would want to increase glycolysis by increasing the activity of these enzymes. Now, you can think of glycolysis as being in 2 phases, and the first phase may surprise you because it's actually an energy input phase, whereas the later phase is energy output. Let me explain further what I mean. At the start of glycolysis, we actually have to put energy into the glucose by phosphorylating it. So 2 ATP donate phosphates onto the glucose. So that leaves 2 ADP coming off. And now by the addition of that phosphates, or those 2 phosphates, the glucose is energized, and that allows the rest of the reaction to proceed, or the rest of glycolysis to proceed. After that initial energy input phase, the carbon molecule, it's no longer glucose, but it splits into 2 3-carbon molecules. So we split a 6-carbon molecule into 2 3-carbon molecules, which undergo further reactions. And those reactions include reducing NAD+ to NADH, and also now we get some ATP out of it. So 2 ADP are made into 2 ATP. And that's happening on both of the 3-carbon molecules that are coming from splitting that 1 6-carbon molecule. So I'll draw those over here. I won't draw the inputs, just the outputs. Now, if you take a look at the math overall, we put in 2 ATP, but we got out 4 ATP. So we had 2 ATP in, I'll write that as -2, and 4 ATP out because we got 2 ATP for each of the 3-carbon molecules that were made from the 6-carbon molecule above. That's a net gain of 2 ATP per glucose. If we look over here at the outputs, we can see that we've got 2 pyruvate molecules. Those are the 3-carbon molecules that are left at the end of glycolysis. So we have pyruvate, and we have 2. I'll write in brackets 2 because that's per glucose. We have NADH, that's a high energy electron carrier, and we've got 2 of those per glucose that we put in. And we've also got out a net of 2 ATP per glucose. We put in 2, but then we got out 4. If we back up and look at the inputs, we put in glucose, and we also put in NAD+, and H+ as well. This is an and sign. So those are our inputs and our outputs. Now, the ATP that is being made here is being made by substrate level phosphorylation. We are directly donating a phosphate from the molecule that is going down glycolysis is no longer glucose over here. I'm not naming it, but we're directly donating a phosphate from that to ATP. So this is being made by substrate level phosphorylation. Key to recognize here is that this NADH contains a bit of energy, the ATP contains a bit of energy, but the pyruvate still has a lot of stored energy in it. Also note that we think of oxygen being required for aerobic respiration and carbon dioxide being produced, but so far we have had no oxygen required. And we've also had no carbon dioxide produced. We're going to see both of those coming up later in aerobic respiration, but so far we haven't encountered them. Looked at another way, if we think of our overall equation for cellular respiration, so far what we've done is we've split glucose into two 3-carbon molecules. Pyruvate, we haven't required any oxygen yet, we haven't made any carbon dioxide yet, or any water, and in terms of energy, there's now a little bit that's been transferred from our original glucose to ATP. There's also a little bit that's been transferred to NADH, that's our high energy electron carrier, and to get that from NADH to ATP, we're going to need to move to the electron transport chain later, so we'll see what happens with that NADH later. But most of the energy is still in the pyruvate. And so we're going to need more stages of aerobic cellular respiration to unlock that energy. So in summary, glycolysis is an evolutionarily old process. We split glucose into two molecules of pyruvate, and it doesn't require any oxygen, doesn't produce any CO2, and actually leaves most of the energy still in the pyruvate. But at least it gets us out a little bit of energy, which is now stored in ATP. Next, we'll look at the Krebs cycle.